I'm so excited. All right. Hi, everybody. I am so thrilled to be able to introduce today's speaker for our CPC seminar. I am here with Mary Playdon, who is joining us from the University of Utah. She's an assistant professor of nutrition and integrative physiology with an appointment in the CPC program at the Huntsman Cancer Institute. She originally did her bachelor's in nutrition and dietetics and her MPH at the Queensland University of Technology in Australia. And then she worked as a clinical dietitian for a while before joining a few research teams here in Colorado, where she worked as a research dietitian on different weight loss studies in cancer survivorship with the likes of Henry Thompson and then Melinda Irwin at Yale. She has a PhD in epidemiology from Yale, and she did her postdoc at NCI. She is a former K99 and current R00 awardee, looking at metabolomics profiles and endometrial cancer risk. And her current research program focuses on diet and obesity in the development and progression of cancer, and the integration of different omics in several ongoing studies, including pilots in timing of energy or meal intake, as well as a UO1 looking at metabolic dysfunction in colorectal cancer risk. So quite a range of things to talk about today, and I'm really excited to hear from you.
Great. Thank you so much. I'm really excited to be back here. I feel like I'm coming home a little bit, and it's really great to see some familiar faces in the audience, so thank you for coming to support me. And thank you for inviting me, Emily. So we have very similar interests, so I'm super excited to actually make your acquaintance. But today I'm going to talk to you a little bit more about some of my earlier work that was focused on the molecular aspects of diet and developing dietary biomarkers. I know that's kind of an interest of yours, and so I kind of focused the talk around that a little bit. But as you mentioned, I'm a nutrition and cancer epidemiologist, and I'm going to tell you a little bit about some of the various work that have gone on over the years and kind of what I'm looking towards in the future. Okay, so does diet matter in the context of cancer? I feel like I'm preaching to the choir with people in this audience because there's such a strong focus on nutrition and obesity research here. But I want to talk a little bit about some of the barriers that we come across doing dietary research and what we can do to overcome those barriers using emerging technologies. How can we really leverage them so that we can actually have a high impact on cancer prevention and control? Most cancer patients want to know when they're diagnosed with cancer, what should I eat? So cancer patients, they're relying on their clinicians, they're relying on their treatment and faith that it's going to work. But a lot of things are out of personal control. But what we put in our mouth is something we can all control. And so I've been told by a lot of the oncologists that I work with that this is the first question they get: what should I eat? And they come to me and say, what should I tell them? And so this is where I think, you know, we have a lot of work to do. And I'm going to tell you kind of where we're at. So what do you all think? Do you think diet matters? I'm seeing a lot of head nodding. So a little bit of background in nutritional epidemiology. We kind of learned that migrants that moved, particularly female Asian migrants that moved from Asia to North America, started to adopt cancer rates of their new country. So for example, women that moved from Asia to the US experienced 4 to 7 times higher rates of breast cancer compared with their counterparts in their native countries. And so the prevailing view is that cancer is a genetic disease, and it certainly is. But these studies kind of showed us, well, we need to start looking at the environment as well. And diet is one of those factors that varies a lot across the globe. And so this is something that really kind of started to stimulate studies in diet and cancer early on. But what really drives my passion for this field is this very simple statistic at the top, that 1 in 2 men and 1 in 3 women will get cancer at some point in their life. So that to me is a very impactful statistic. But what really stands out to me is that over a third of cancers, almost half of colorectal cancers, up to 60% of cancers of the uterus are preventable. And so that really motivates a lot of my research. What about after a cancer diagnosis? So when we think about cancer patients going through treatment, we think about weight loss, malnutrition, undernutrition, and that's definitely a challenge. But overnutrition is also a challenge with cancer survivors, especially after the treatment period and looking towards how obesity and overnutrition affect survival rates. So in 2022, the NIH convened a workshop, the Pathways to Prevention program, to really hone in on what are the knowledge gaps and what should we really prioritize. And their report is really eye-opening. So there are just enormous gaps in testing rigorous nutrition interventions in cancer patients during treatment and throughout the survivorship trajectory. So designing and testing these interventions, but also when should we intervene? We don't know when is the best time to intervene. And so those are some priorities for cancer survivors. That's something that my group is focusing on also, with a particular focus on malnutrition, but I'm not going to talk about that today. Just the fact that there's a lot of work still to be done in the cancer survivorship space. I'm going to talk a bit more about cancer prevention today. So in summary, the research I'm going to talk to you about today is really investigating the interface of diet with human metabolism and cancer. So we want to identify the metabolic pathways that are dysregulated years before cancer initiation, because we want to intercept cancer before that initiation stage. But then also among patients that develop cancer or experience cancer, how can we use diet strategies to help them to live longer, better, healthier lives, considering they're living with cancer. So this is something I just like to talk about and acknowledge. So who here has been confused by nutrition messages in the media? So I really like this example because all of this happened within the span of about a year. First, we hear laws where we need a cancer warning on coffee. Then, oh, okay, we're going to retract that. And then large studies coming out saying, oh, actually, coffee protects you against death from all causes. So it's very confusing. Some of the confusion is perception, but some of it is real because of, and any of the epidemiologists in the crowd will know there are methodological challenges. So sometimes, the results we're putting out can be impacted by some of these challenges. The problem with this is that the public begins to distrust nutrition research, and then they distrust diet guidelines. And that's not what we want. We need to build trust in what we're telling people to do in terms of diet. So that really motivates me as well. So we actually do have quite convincing evidence for a number of diet exposures in terms of what increases or decreases cancer risk. And you can see some of that summarized on the left, but there are still a lot of questions. And some of my earlier work, which I'll talk about today, is really addressing that first point, imprecise diet measures. So typically in epidemiology or in large-scale population studies, for cost effectiveness, we rely on questionnaires, self-report. So who here can remember what you ate 12 months ago? I definitely can't, but that's the premise behind traditional diet questionnaires. And I'm not bagging traditional diet questionnaires. We need them, and they've really contributed to the foundational links between diet and chronic disease, and they've contributed to evidence-based guidance for prevention of multiple chronic diseases. It's very important, but there are some issues with measurement error that can sometimes, there may be a real relationship, but it makes it difficult to see, especially in the context of cancer, because the magnitude of the relationship is not massive, like smoking and lung cancer, for example. And so you need precise measures if you're going to really be able to see those relationships. The other thing that we've had challenges with is, looking at cancer subtypes. So even a tumor that arises from the same organ can be completely different at the molecular level and can respond completely differently to diet. So traditionally, we might look at all breast cancer together. That's just an example. But hormone responsive and, negative tumors they have a very different relationship with diet. But now we need to really move towards these very large consortium approaches to pull data so we can really look specifically at tumor molecular subtypes. And other issues that are listed here that I won't go into, but there's a lot of challenges. I would argue that studying diet is the most complex exposure that you can possibly study because a dietary pattern, you're not just exposed to a collection of foods, it's actually tens of thousands of chemicals that your body is exposed to, and that's what we're trying to study. And so in some of the early work that I did in my PhD with Steve Moore at the NCI, it was really at a time when, you know, metabolomics has been used for decades in some sectors, like the environment and environmental epi, for example, but not so much in public health. And so he was tasked at the NCI of bringing metabolomics and applying it to cohort studies at the population level. So it was really cool to be there at that time to kind of see this initiative take flight. And we used metabolomics data from some of the NCI cohorts to start to identify diet-related molecules. And that was kind of some of the beginnings of nutritional metabolomics. So there's chemicals in food, and we can detect them in your blood, in your urine, and in other tissues. We can detect molecules that are derived from diet, that are digested and absorbed and broken down. They're metabolized, and then we can also detect your body's endogenous response to eating certain foods or diets. And so we're capturing all of that if we do like an untargeted metabolomics run and relate it to self-reported diet. So that's what we did using data from various cohorts, just looking at associations between foods and blood and urine metabolites. And in general, we picked up markers, hundreds of markers for different foods initially, and then we started to replicate it across different cohorts with different population characteristics. And it was really nice to start to see the replication. This is just an example of some of the molecules that, you know, we detect in blood or urine that are diet related. And so it's kind of, you can see that it's capturing a fairly good breadth. We've got fruits, vegetables, we've got, you know, diet supplements, beverages, protein foods, fat sources. So you can capture aspects of the whole diet. And you can capture it in as little as, you know, 50 microliters of whatever material it is. So you don't need to be able to read that, but we were able to validate those observational findings in a really unique feeding study of habitual diet. So when you think about feeding studies and diet intervention studies, traditionally you think about testing diet A against diet B. So you have a major comparison that you're looking at. This was unique because it was an ancillary study within the Women's Health Initiative, which is a large long-term cohort. We partnered with Marion Newhouser and Joanna Lampy at the Fred Hutch on this study. Let me go to the next slide because it gives you a little schematic. So 153 women did this feeding study. They measured diet before the study with weighed food record. And then the metabolic kitchen prepared those diets and fed it back to those women over 2 weeks. So instead of comparing 2 diets, we're comparing 153 diets because each one was individualized. And at the end of the 2 weeks, they took a fasting blood draw and they collected 24-hour urine samples. So what was really unique about this study, a little bit difficult to see, but on the left, you can see distribution of food intake for certain food variables. And you can see some are more evenly distributed, others are very skewed. This is what we see in population studies. And so this is recapitulating what we see in the population, but just with much better diet measurement. Yeah, so it's very relevant to the real world. So what we found with this study, detected biomarkers for 23 distinct foods, beverages, or supplements, and 171 distinct diet-related molecules. what really came out on top in terms of the strongest correlations were foods that are consumed habitually, like coffee, for example. People tend to drink it every day. So even if a marker has a short half-life, you're still going to be able to pick it up in a blood sample. So it makes sense that those would come out on top. But when we're thinking about diet at the population level, we're interested in those foods that people are eating regularly, because that's likely to have an effect on chronic disease, rather than something that's eaten once a month. You might not capture it in your blood, but how much of an impact is that having? So these things that you're consuming all the time is what we're interested in. So what about blood versus urine? We compared these by specimens. A lot of cohort studies store blood, but far fewer store urine. And in the observational studies that we did have data for, we were detecting a lot more molecules in urine. There are a lot of conjugated excretion products, which makes sense. But we were interested in this feeding study scenario, where we're going to see the same thing. You can see the magnitudes of correlation are kind of similar between serum and urine, except for maybe a couple of groups, like dairy seem to be better measured in urine, actually. But otherwise, the correlations were comparable, so that you can be fairly confident if your cohort has blood or if it has urine, you're going to still be able to pick up good biomarkers. But not only can we capture what people ate, we can tell how the food was prepared and processed. So this is an example of that, looking at urine-related coffee metabolites, so hierarchical clustering heat map. So you can see how the molecules kind of cluster together. I think caffeine metabolites are in the top right. There's polyphenols kind of somewhere towards the middle. And then there's big cluster of molecules that are all related to flavor compounds, aroma compounds, and kind of roasting metabolites. So we can tell if you drank dark roast coffee because your levels of trigonelline will be much higher. So as opposed to asking a question on a questionnaire, do you drink coffee? How often do you drink it? Now we can get all of this information from a drop of your blood, which is pretty cool. And to put it in, sorry, these are kind of hard for you to see, but I'll talk you through it. To put it in perspective with, what we're seeing in population studies, we can look at the magnitudes of association. They're much higher because we have better diet measurement. But it's telling you really what is the kind of true correlation there. And I just want to put it into perspective, because I hear that you're using doubly labeled water and things like that with some of your interventions. Gold standard biomarkers, which have a correlation, energy intake in doubly labeled water of about 0.6, 0.7. And we're seeing correlations close to 0.9. So these are robust biomarkers. To put it into perspective with what's being considered gold standard in nutritional epidemiology, since we started looking at biomarkers. What's really exciting for me is the amount of data we have now that we can start to do nutritional epidemiology analyses. So the consortium of metabolomic studies was started by my NCI mentor, Stephen Moore. So it was really cool to see it get started from the beginning in about 2014 is when it started. But now we have representation from more than 70 cohorts across the world that all have blood metabolomics data, they have phenotypic data, and have disease outcome data. So they're all part of this consortium. And if you're interested, you can become an ancillary member of the consortium if you're not contributing data, and become a member if you want to contribute data. It's not sharing data, but they've actually developed a full analytic pipeline, which is publicly available, Comets Analytics, where you can share summary estimates, and then NCI will meta-analyze it for you. So they have like a standardized input template. You work with each cohort to get the input template right. So essentially, everything is done remote. And then you can do these big, large-scale analyses through this Comets Analytics platform. The table on the left is just kind of showing you sample size of people of the cohorts with blood metabolomics and dietary data. That was in 2019 when the protocol paper was published. Now we easily have double the sample size. So it's very big, which is cool. So I'm working on a COMETS project right now with Emily's prior mentor, Fred Tabung at Ohio State University. We're doing a multiple cohort meta-analysis looking at dietary patterns of colorectal cancer risk and how that relates to blood metabolites. So we're currently it is actually quite a massive endeavor because you have to harmonize all the diet questionnaires. It's a lot of work, but it'll be a resource for the community down the road. But if anyone's interested to join, just email me because I can show you where to go to sign up. So I also collaborated with an international team just to look at, okay, what are biomarkers with the highest level of validation currently? We kind of worked on this over several years. This was just published a couple of weeks ago. Ricard Landberg and Augustin Scalbert were the kind of primary drivers of this. And then we worked with Erica Lockfield at NCI and other folks like Inge Heibrechts at World Health Organization. We did this massive review. So if we want to look at, how valid is a biomarker for population research, we need to look at these validation criteria that are listed in the top right. Here is a list of candidate biomarkers with the highest level of validation based on these criteria. It's not that big of a list compared to our master table, which literally had hundreds of biomarkers. So I think we're off to a good start. We've identified a lot of molecules, but we need to validate them. And I know that work is going on here with feeding studies, and actually that's exactly what we need. So the one thing that really stood out was this time response validation criteria, looking at the pharmacokinetics and pharmacodynamics. We need more studies to look at those aspects and look at, inter-individual variation and how that affects the biomarker levels, because that's really important as well. So this was a European consortium, Food Bowl. I think it was a play on football because they love football in Europe. So it was 22 countries contributed, and they did 2-week feeding studies systematically working through diet exposures to validate diet biomarkers. And that information is curated here in the Exposome Explorer database that was developed and is kind of housed and run by International Agency for Research on Cancer as part of the WHO. But they've also curated they curated information from the literature, from other population-based studies. There's a lot of information on here if you're interested in diet biomarkers. They also fairly recently curated information on microbial metabolites. You can download nice Excel files full of all the literature on microbial metabolites. For example, if you do a panel in whatever study you're doing and you want to find out more information on these molecules, and is there some role to the microbiome, it's kind of a nice resource to check against. So moving a little bit more towards cancer, when I was at the NCI, we wanted to kind of do proof of principle analyses. Obviously, we were kind of identifying these biomarkers. They weren't validated yet, but we wanted to in proof of principle, look at the relationship of diet-related metabolites and risk of cancer. And so we first did this looking at incidence of breast cancer in the PLCO cohort, which is one of the NCI cohorts that followed people for decades to see who got cancer. They took a blood sample and did a food frequency questionnaire at the beginning of the study. And the blood was taken on average, I think, 7 to 8 years before cancer diagnosis. So we had data on about 1,300 women, cases and controls that were nested within this cohort. And we categorized about 55 diet exposures we were interested in to look at. And we kind of did a 2-phase analysis, first looking at the diet-related metabolites, and then of those, there ended up being about 113 molecules that were related to diet, looking at the relationship with breast cancer. And so here you can see some of the results. I'm just showing you total breast cancer and ER-positive breast cancer. We were a little bit underpowered to look at estrogen receptor negative breast cancer, which it's difficult because it's more rare. But here what you're seeing is odds ratios and confidence intervals for the various metabolites and what diet exposures they were associated with. So the three in bold were significantly associated with total cancer, and then this whole panel was significantly associated with positive cancer. So you can see that when you split the subtypes, you get far more hits than if you're putting the cancer types together. So what it really pointed towards was alcohol being a risk factor for breast cancer, which we knew, but this pointed towards mechanistically why. So it really pointed towards sex hormone metabolism. Then it also pointed towards specifically dairy fats, like that caprate molecules, so specifically a dairy fat, what you would find in butter, for example. And this is a controversial area for decades, is fat related to breast cancer, but maybe it's very specific types of fat that we need to be concerned about. And then vitamin E, it was really interesting. So the type of vitamin E found in frying oils increased breast cancer risk, but the type of vitamin E in supplements, which is alpha tocopherol, was protected. So that was kind of interesting. So it really focused on these three kind of diet exposures and pathways. So with metabolomics data, you're linking to these databases with molecular information, but oftentimes there's a lot of missing information. So sometimes using a data-driven approach, you can get a sense for biological mechanism through leveraging the relationships of the molecules in your data set. So this is an example of a type of analysis where you're kind of looking at the conditional correlations between molecules, conditioned on the presence of all the other molecules, where the lines represent correlations, pink being positive, purple being inverse. And you can see how molecules start to cluster. This is actually kind of useful if you have unknown metabolites. You can still include those in the data and see how they end up clustering. So something might be kind of not fully characterized, but it might be a very significant, have a significant association with your outcome. You can see where it sits in terms of how it might interact with known pathways. We were also interested in obesity-related metabolites and risk of breast cancer, because obviously obesity is an important risk factor, especially for postmenopausal breast cancer, and this was a postmenopausal cancer data set. And so we did find a couple of pathways of metabolites within these two pathways that were significantly related. So sex hormone metabolism, which we would anticipate, but also degradation of amino acids. So when this pathway is dysregulated, the molecules in red accumulate, and that's what we were seeing in patients that ended up getting cancer. So branched chain amino acids are it's really interesting. These are turning out to be important in the etiology of diabetes and other things, and so that really has gotten me interested in the interface of metabolic health and obesity-related cancers in general. So when I transitioned to the Huntsman Cancer Institute, I wanted to continue working with obesity-related cancers, but kind of separate from my mentor a little bit. I was still interested in female cancers. Endometrial cancer, or cancer of the uterus, is it's a leading cancer type in terms of incidence. And the rates, if you look on the bottom right panel, the rates have been going up quite significantly over time. Not just the more obesity-related, hormonally driven endometrioid subtype, but the other subtypes too. And in particular in racial and ethnic minority groups, like why are rates of this cancer in particular going up? Partly obesity rates have been going up, and this is probably the most obesity-related cancer. But there are other factors that we need to investigate. So we wanted to look at metabolic dysfunction and risk of endometrial cancer. So I did this with a similar approach to the breast cancer study, except now we were looking at data from 3 cohorts and then doing a big meta-analysis of the data. So endometrial cancer is a little bit less prevalent than breast cancer, so we have less samples to work with, but we did have close to 900 samples overall. And we ran metabolomics on blood samples from the PRCA cohorts, but also the Harvard nurses health studies. Might be a bit hard to see, but we wanted to look at BMI independent and possibly BMI driven metabolites. So we first ran the analysis while adjusting for body mass index, and there were 80 metabolites that were nominally related. It wasn't significant once we had accounted for multiple comparisons. I think we needed an even bigger data set than we thought we needed. And I think this is the case. That's why the Commons Consortium is so critical, because you just need really big samples. But nonetheless, it kind of points towards pathways that might be interesting. And I kind of summarized them up here. And they're kind of molecular pathways we would expect to be linked with endometrial cancer. Inflammation, oxidative stress, for example, advanced glycation, things that are relevant for diabetes too. Branched chain amino acids pop out again. Gut microbial metabolites, there were several of those which I thought was interesting. That would be interesting to pursue. And steroid hormones. And we did a metabolic mediation analysis, so we're looking at how much of the relationship between obesity and endometrial cancer could be mediated by some of these metabolites. And we ended up finding some significant hits, and they kind of point to similar pathways actually, especially insulin resistance, oxidative stress, and inflammation, which are all important cancer pathways. But these are things that really obesity is probably promoting to drive endometrial cancer. So the World Cancer Research Fund and American Institute for Cancer Research summarizes all the literature to date on diet and cancer risk, and they have some reports for cancer survivors. They do they continually update the information, but it's these large groups doing these big systematic reviews of meta-analysis. But for endometrial cancer, you can see that a couple of diet factors, that there is some pretty strong evidence that coffee drinking is protective, and a high glycemic load diet likely increases risk of endometrial cancer. So this is pointing to inflammation and insulin resistance. So these are two pathways that clearly are important. So I mentioned my collaborator at Ohio State, Fred. So he's developed these kind of empirically derived dietary patterns, a dietary pattern linked with inflammatory biomarkers, and a dietary pattern linked with C-peptide, which is a marker of insulin secretion. So kind of looking at the inflammatory potential of diet and the hyperinsulinemic potential of diet. And you can compute these dietary pattern scores, which is like a single continuous variable. And it's essentially a variable that's accounting for pro and anti-inflammatory foods, hyper and hypoinsulinemic foods. This is a lot of info, but this is how the algorithm is computed on the top right. So he developed these dietary patterns in the nurses' health studies, and he validated them in the health professionals follow-up study. You can see the kind of hyperinsulinemic and hyperinsulinemic foods. Some things you might think are counterintuitive when you think about healthy versus not healthy foods. But this is what the data are telling us is impacting these particular molecular pathways. And it's similar for the inflammatory pathway. So you can see the pro-inflammatory and the anti-inflammatory things. So alcohol and coffee are actually anti-inflammatory. This is a very sensitive thing when we're talking about communication for cancer prevention, because we want to advise not to drink any alcohol. So this is something that I know Fred has had to be really sensitive about in grant applications and things like that, not necessarily trying to promote alcohol drinking, but it does affect these metabolic pathways. So when we looked into our data set and the relationship of these dietary patterns, we see markers of foods that are incorporated into the algorithm, which is kind of telling us, yes, we're measuring the right diets, but then also these endogenous pathways that are associated with eating that way. And coincidentally, a lot of these in the endometrial cancer analysis were related to endometrial cancer. So what we're doing now is replicating these analyses in data from colorectal cancer patients. We did compute a metabolite score to look at endometrial cancer risk with the metabolites, but we were a little bit underpowered. We ended up publishing a paper. We looked at the dietary patterns in relation to endometrial cancer risk using all of the nurses' health study data, so hundreds of thousands of women, and it showed a significant relationship of both diets with endometrial cancer. So I'm really interested to do some diet interventions using these dietary patterns specifically with endometrial cancer patients. As kind of a next step. You hear all the time for cancer prevention, eat a plant-based diet, and within the concept of a plant-based diet, we're also interested in does the botanical diversity that you choose to eat matter? Like, does eat the rainbow matter, or can I just eat broccoli because I only like broccoli? So Henry Thompson at Colorado State University did some work on this like many years ago. In the 2000s, he ran a trial actually looking at high versus low botanical diversity given the same high fruit and vegetable diet, and he did show significant differences in DNA oxidative damage and lipid peroxidation. So we developed an algorithm to score botanical diversity of the diet. So we can take a food frequency questionnaire, and using this metric, which is like a Simpson index, which is used to look at microbial diversity and microbiome studies. So we can look at botanical diversity. So in our data sets, we saw a lot of plant-derived molecules related to the diversity of botanicals in your diet, which is what we would expect, but then a lot of endogenous pathways too, which I think is really interesting. So again, we're looking at this in colorectal cancer patients. We're also looking at gut microbiome characteristics in relation to botanical diversity and gut symptoms in colorectal cancer patients. So just to wrap up, I've been talking quite fast. It's a lot to cover. But what about in the cancer, so in the cancer patient, cancer survivorship space? And also, how do we take all of this enormous amount of data? Like, what do we do with it? Also with the understanding that two people eating the same diet are going to respond differently. So how do we personalize diet recommendations? And how can we leverage all of this nutritional metabolomics data, accounting for the fact that people have very specific molecular subtypes of cancer. They're on very specific treatments, especially now. Treatment is moving towards very targeted precision therapeutics. Diet can significantly augment the efficacy of cancer treatments. This was a really nice kind of proof of principle study that was published in Science that showed that eating a high fiber diet significantly improved progression-free survival in melanoma patients getting immunotherapy. And so it's a relatively simple recommendation. We definitely have ways to increase fiber in the diet that can significantly impact treatment response and survival. So this is kind of what I'm interested in. What are the dietary tweaks that you can do to help cancer patients have better outcomes? And considering one size doesn't fit all. So I was really intrigued by a group at Imperial College London that used they curated information from some of these metabolomics databases, and they used artificial intelligence algorithms to predict the anti-cancer potential of various food-related chemicals. And they kind of built a map of anti-cancer fighting foods. So you can see some of the, sorry, I replicated some of this, but things like, various fruits, onions, garlic, citrus, carrot, celery, coriander, turmeric, dill, had a lot of cancer fighting chemicals according to their algorithm, which sounds a lot like my Thanksgiving turkey stuffing recipe. So don't feel bad at Thanksgiving if you want to eat the stuffing. I was chatting with Henry Thompson about this, and just really fun. We started thinking about, well, could we do something similar but make it a little bit more specific to the tumor type and the cancer treatment? Like here's kind of like a vision of, let's focus on protein kinases that are really targeting specific mechanisms as a treatment. Then let's pull information from all of these databases, which is publicly available. Let's put it together and let's build a database where you can query a particular tumor type and a particular drug, and you can overlay it with food molecules that have similar activity on the gene targets of interest, and build precision dietary patterns that way. So we're doing that now. So we've started a collaboration with biomedical informatics, and we're currently building this connectivity map where this is kind of the process, where we're ranking food metabolite relationships, we're ranking drug gene activity relationships, and then we're building this connectivity map between them, and then also at the chemical level, looking at chemical similarity using chemical similarity enrichment analysis to look at similarity of the molecules, and then using AI to build precision dietary patterns. So that's in the works. It's really fun learning to speak a totally different language, working with the bioinformaticians, but it's a lot of fun. This is just an example, although this is super simplified, but being interested in type 1 endometrial cancer, let's think about a target, mTOR, and a drug that works with it, metformin. Let's look at chemical similarity, like certain polysaccharides maybe you could build into your diet. So let's build a diet with those components, and then let's test it. That's oversimplified because I think at least the literature is showing that when people take metformin, might matter in terms of outcomes. So this is more conceptual at this point. But the goal is to work with our brand new metabolic kitchen at the University of Utah that we built. We hired an RD to manage it, and we hired a chef to make it all taste good, so we can start to build precision diets and do trials with cancer patients. So just coming back full circle, I think we're in a really exciting place to build on the guidelines that we have right now for cancer prevention and for cancer survivors. It's an exciting time thinking about diet and what it's doing at the molecular level and how we can personalize it. So I'm excited. And I'd just like to thank all the people that I've worked with and my team. Happy to take any questions.
So I've been to metabolomics and biomarker talks for the last 20 years. I thought this was the most interesting one I've been to, especially the first part where you can identify broccoli, things like that in the diet. It's probably, you guys are all familiar with that. I'm a nephrologist. I thought that was fascinating. My question is, there was a lot of sort of natural foods that you were able to identify, coffee, tea, broccoli, et cetera, but I didn't get a sense that there was a biomarker for like Cheetos and the processed foods, and maybe I missed that, but
yep.
Are there biomarkers for eating at McDonald's every day and Cheetos and
no,
all
this
those?
is exactly what Emily's working on. So, right?
Some of that, yeah.
So I know there are folks that are working on this. Part of the difficulty is even defining what processing means, because there are different levels of processing, and there's a lot of controversy over how to define it. So we need to get that right first, and then we can start to identify what the biomarkers are. I don't know if you want to speak more to that.
I actually had almost a related question, and it's more about how we measure the foods that we're eating currently. And so I noticed in some of those earlier slides too, you see those individual foods, and then like snack foods was a category, right?
Yeah.
And so it's like, how do you dump all of those foods together when
Yep.
they probably are really.
So
doing
that's
unique
why we
things.
need feeding studies too, because the cohort studies that we have data and samples and follow-up disease outcomes based on questionnaires that summarized average population diets, whatever the population was. And it groups things like snack foods into a single category, which included like chips, popcorn, cookies, whatever, with very different profiles. So we need to devise different questionnaires and probably do the feeding studies where you're specifically testing certain foods, like energy drinks. What is that doing? Nobody measured any energy drinks in most food questionnaires, and like a lot of these milk substitutes, thinking like impossible meat. I mean, so many products now that were just never measured previously. So there's a lot of work that still has to be done.
Can that be done in mice? Can you just give?
Yeah.
Yeah.
But that's a really good point. They've started to look at ultra processing as an exposure in itself and links with chronic diseases, and it's turning out to be really important. So
Thanks for that talk. That is just fascinating. I'm just fascinated by your work. As you start to gather data from like different places and things like that, where you might use 5 or 6 different metabolomic scores trying to gather data.
Mhm.
How big of a problem is it with the variability that comes with collection, processing, their machines, their sensitivity, how they interpret? When we tried to do this at the center level of getting different metabolomic scores on the same page, it's really challenging.
completely agree.
So how has it been getting to where you have been,
Yeah, so
where you are right now?
let me talk about comets because they deal with this problem. There are probably 5 major platforms that cohorts go to, like Metabolon, Broad, Nightingale, Biocrates. Trying to think of something else. They're the major ones, but they are still all different. So the NCI, the IMS group, the IT folks at NCI, they have a metabolite harmonization process that they've worked out. So they have like a kind of a harmonized chemical, I guess, variable name. You know, metabolites can have 20, 30, 50 synonyms. And they're actually working, they're like scraping through all of that data and harmonizing it. They worked predominantly with, I guess there are some maybe targeted, but a lot of kind of semi-targeted platforms. But yeah, mixing targeted platforms, concentration data and relative intensity data. And then that's challenging in itself. The way Comets is approaching it is doing cohort specific analyses, and then because you tend to be comparing cases and controls, as long as they're analyzed, as long as the samples are collected the same way between cases and controls and processed the same way, then you still get the case to control comparison within that cohort, and then you can meta-analyze across the cohorts, but you would not pool the data. You couldn't really do that because of all these issues. And the sample handling. So Steve Moore published a paper on effective sample handling on blood metabolites. And they did, I can't remember how many different comparisons of freeze thaw time and et cetera, et cetera. It did matter, especially for lipids. But as long as you're handling cases and controls the same way, you got similar results in the end.
I have a question. Is there any difference between like organic and non-organic food? Between like all these like vegetables?
I am not aware of organic versus non-organic studies. Are you aware of any?
Our team here, so if you guys don't know, like Nicole Reisdorf runs the mass spec lab out of the School of Pharmacy. And so we have some foodomics work out of that group. And we looked at bell peppers, for example, and looked at things like country of origin, organic versus conventionally raised, and color to see which factors would distinguish between metabolomic profiles. And they just looked at the lipids, so I guess it's just lipidomics, but they found that conventional versus organic wasn't different, but color, for example, did distinguish between the different pepper types. I don't know if that's done regularly because, you know, generally looking at organic versus conventional is one thing, but even looking at different food types, dietary patterns, there's just not a lot of work being done in that space that I know. Something that I think a lot about in this work is what's the level of precision that you really need with this sort of biomarker work or even in a precision nutrition intervention? Because, you know, especially on the diet biomarker side, I get questions all the time, like, do I really care if you're eating like an organic red bell pepper or is it just like vegetables? You know, like translating what we're able to measure to the messaging that we give to patients and survivors or just the general public. And I'm interested in your thoughts on that.
I mean, in terms of the research, it depends on the research question about how much precision. But I hear what you're saying about really when you're distilling the messages at the population level, you want to keep it simple. So I don't know. So far in the analyses that I've done, because I'm working with data that a lot of it is already grouped and it's not as precise. That's where we need, better design studies that way. I've come across this problem looking at the botanical diversity because you need to be at the granular level to look at botanical diversity. And most cohorts group green leafy vegetables together, orange and yellow together. Like there are certain groups that are just grouped together, but botanically they could be very different. And this has been a real problem because we're harmonizing FFQs across cohorts. Some of them have a lot of grouping and some much less grouping, but you can't compare them really. So if you're looking at botanical diversity, you couldn't really compare the cohorts just because of the way they group their variables. You could only make a conclusion based on within cohort analysis. So yeah, I guess when it comes to those types of questions, then more granularity is better. Yeah.
I have a question related to the very intricate feeding studies that you did and presented in the beginning. And you mentioned during your presentation that people respond to different intakes differently. Were the studies that you performed that were very precise, were you able to disentangle, like in certain groups, this metabolite was a great marker and it wasn't a great marker in another group? Or did you not have that sort of variability in the population in the studies?
We looked at effect modification by a bunch of different variables. So, and it wasn't a major thing. So we looked, I think we looked at age groups, we looked at different BMI categories. I'm trying to remember what we looked at. Yeah, but ultimately nothing was really significant. We were looking at effect modification. So like most of the markers should work in various groups. But again, it is a homogeneous population. So this type of design needs to be replicated like in men and in, other more diverse populations.
Or even like food metabolism genes or something like that could vary.
Yep. But
that you have to know what to look for, I guess. Yeah. when you're doing the effect modification studies. Yeah. Yeah.
So the Fred Hutch group is I'm forgetting what the consortium is called, but they were funded along with the Harvard group, Duke, and various institutes to kind of do food ball in America, but kind of taking on foods that food ball didn't look at, but also with a focus on looking at inter-individual variation. I think that's a real focus of it. So they're doing that right now.
Yeah. I think nutrigenetics is fascinating.
Yeah.
So this is maybe kind of a weird question. Do you have a sense of how much variability in the metabolites can be explained by what you eat? I'm just thinking of like a newspaper headline. What percentage of are we are we, you are what you eat type of thing? Like
I really love that
versus
question.
something that's necessarily like an innate, you know, that you can't do anything about, but the nurture nature kind of issue, more broader issue.
I don't think I have a percentage, but that's kind of a really interesting question, especially if you're kind of comparing it to other things. To think about that. So it could be a really interesting analysis.
So I was just thinking about all this variability. And so I'm curious about another source of variability, and that would be like cumulative diet, as opposed to like, a lot of this was kind of snapshot diet,
Exactly.
right? How does that fit in? Because I'm actually thinking about you and Henry and your little plan there. So how, and is there a role for cumulative diet, and how would that fit into that?
Yeah, I would say there probably is a really important role. I know that here you know, at CU here, you're doing a lot of work on these longitudinal cohorts and looking at exposures across the life course. And honestly, we need more research in that area. I don't think we can really tell, you know, first of all, when are the important exposure periods in relation to the various chronic diseases? Like, when does it actually matter? I don't think we know that yet. And then this idea of cumulative exposure. I guess I'm aware of some studies that have looked at longitudinal measures, but it might be over a year or two, but not really longer than that. So I know that would actually be kind of interesting as a question in some of the cohorts that do have routine diet assessment over time, like the Nurses Health studies actually do every X number of years, they do another FFQ. So if you kind of look at that question.
I'm just kind of thinking some of the latency periods for some cancers, right?
That's
So
true, yeah.
are we measuring the right thing, right? And
Yeah, no, that's true.
just more variability for you.
It's true because you don't know, you're getting people at a certain point when they enter a study, but where is that in the trajectory of cancer?
Exactly.
You know, some people get cancer a year later, some people it's 20 years later. So you're capturing them at different periods in cumulative exposure. Yeah.
And I would say it's similar to what you just said is how much does that snapshot of the diet that we're collecting now represent what they were eating 10 years ago? I mean, how stable in terms of the major foods that we're looking at, how similar are our diets over time? Do we really change that much? I can imagine kids do.
Yeah.
Yeah, but once you hit adulthood, I don't know. I mean, because that's, again, it's so expensive to collect this data longitudinally. And if you could be certain that this is a pretty stable biomarker, or this represents diet that probably didn't change much, that would be so helpful to us in interpreting our results, right?
Yeah. So do you think your cohort has that kind of data? You're following some of the adults, right?
Sure.
Lead program. The lead program. I'm just thinking about longitudinal. Oh, no.
We are following adult women.
Yeah.
Mhm.
I don't think we have that kind of data.
Not diet.
Yeah,
No,
we don't.
children.
Yeah.
Oh, yeah, she's on.
there.
Yeah. That on.
Oh, yeah. Maybe this is something that you could address.
Yeah, I think moms is the longest one, right? And we do check diet.
Oh, and then actually we do have a DPP cohort as well, but if they're looking at that like micro data.
Yeah, it might be more broad, but yeah.
Yeah. Definitely a question to think about. Yeah, those kind of data are so unique. There's only some cohorts that capture longitudinal, especially bias systems. Great.
Thank you.
Thank you.
Thank you.